Today we will talk about what is genome, what is hybrid, and what is pure line. So what do you mean by genome? Genome is simply the complete set of genes that an organism carries in its DNA, that is its genome. So we can say that it is a complete set of genes that an organism carries in its DNA. What do you mean by genome? It is the complete set of genes or the complete set of genetic information present in an individual. That is what is called as genome. For example, we say that we have this is an individual it is made up of cells. This is an individual, and this is an individual, and this individual is made up of cells. This is nucleus, and these are chromosomes in the nucleus. These are chromosomes. Half chromosome from the mother and half chromosomes from the father. So this chromosome, it would be like this. This is the DNA. This is the DNA molecule, DNA in a chromosome. So it would be like this. Chromosome. So we can say that the total set of genetic information in an individual, that will become its genome. A complete set of genes which are present in an individual, that is called as its genome. There is another definition for the genome, that is it is the total amount of genes which are present in a single set of chromosomes, that is in a haploid gamete. So also we can say that genome total number of genes present, total number of genes, whether they are expressed or not expressed, present in haploid gamete. This is also it is genome. Or we can say that another definition of the genome is, or we can say that the total number of chromosomes present in the haploid gamete, that is also it is genome. Number of chromosomes in a haploid gamete. For example, we say that in human genome there are 23 pairs of chromosomes. Human genome, there are 23 pairs of chromosomes. Human genome, we can say that there are 23 pairs of chromosomes, or we can say that there are 23 chromosomes, 23 chromosomes in haploid gamete. That is, it can be sperm or egg. So what do you mean by genome? Genome is simply the total amount of DNA present in an individual, that is, it is genome. Or we can say that the total number of chromosomes in an haploid gamete, that is, it is genome. Now sometimes we say that single set of genome, single genome, and this single genome is present with one N. It is present in haploid gamete. This double genome, two N, it is present in diploid cell. This gamete is a haploid cell. Or we can say that triploid genome present in triploid cell, or many genomes present in polyploid cell. Many genomes present in polyploid cell. Sometimes we say that hexaploid, it is having 6 sets of genomes. Yeah, a tetraploid, 4 sets of genomes. We say that what is genome? Genome is simply the complete set of genes which are present in an individual, or we can say that the complete set of genes contained in a haploid gamete. Or we can say that what is genome? It is the total number of chromosomes in a haploid gamete. This is what is called as genome. This is, for example, this is an individual, half chromosomes getting from the father, half chromosomes getting from the mother. This chromosome, it is made up of DNA. This is DNA, and this DNA then controls the different characters. For example, it controls the height, it controls the eye color, it controls the shape of ears. So this is what is called as genome. Then we have got hybrid. What do you mean by hybrid? What is hybrid? This hybrid is an individual produced by the cross between the two genetically different parents. So hybrid is an individual produced by cross between two genetically different individuals, different parents. You can see that. So hybrid is that individual which is produced by cross between two different parents. For example, we have two parents. This one parent, it is having what it is having. For example, we can say that red flowers. This is having plant with red flowers. Obviously, this is red color, red flowers. The genotype is capital R, capital R. Now we'll cross it with a plant that is having, suppose, white flowers. This is white flowers, plant with white flowers. Obviously, it is a recessive character. Then we will get hybrid. This is hybrid. So plant with red flowers. So hybrid is an individual. This is hybrid for a character. This is heterozygous for the trait. The trait is color of flower. So we can say that, for example, we have two parents. These two parents are genetically different. This is capital R, small r, small. And then we'll make a cross between the two different parents and we get a offspring, we get an individual. This individual, which will get a cross between the two parents which are genetically different, that is called as hybrid. This hybrid is, sometimes we use the term heterozygous for the hybrid. Heterozygous for hybrid. So hybrid is an individual. Hybrid is an offspring, or it is a progeny which is having, or we can say that another definition. The hybrid is a progeny, or hybrid is an individual which is having two dissimilar alleles for a trait. So hybrid has two dissimilar alleles for a trait. So hybrid has two dissimilar alleles for a trait. So what do we mean by hybrid? Hybrid is an individual which is formed a cross between the two genetically different parents, and this hybrid is an individual, or hybrid is an offspring, hybrid is a progeny which is having two dissimilar alleles for a trait. Sometimes we use the term heterozygous for the hybrid. This actually this hybrid, it is having the desirable characteristics. That means this hybridization technique is mostly done in case of self-pollinated plants. why this hybridization technique is done. For example, we have got two plants. This is one plant, this is another plant. These are genetically different. They belong to same species, but they are genetically different. Now what do you want? We want that we should get a progeny, we should get an individual which should be having characteristics, we should be having the characteristics from both the parents. And then we make a cross, we take the pollen grains from this plant, then this is these pollens on or dropped on this plant. So this is what is called as hybridization. The progeny which we get, that is called as hybrid. And this hybrid, it is having better characteristics as compared to that of their parents, because of the fact that we get the genes from both the parents in the hybrid, and obviously it would be having superior characteristics as compared to that of the parents. This is what is called as hybrid vigor. That means the hybrid individuals are better or superior as compared to their parents. This is called as hybrid vigor or heterosis. These hybrids can be superior to their parents in one or more characteristics. So hybridization is a technique to get the hybrids in case of self-pollinated plants. So hybrid is an individual which is having two dissimilar alleles for a trait, or we can say that hybrid is an individual produced by the cross between the two genetically different parents. This we sometimes use the term heterozygous for the hybrid, and hybrids are mostly superior as compared to that of their parents in one or more characteristics. This is what is called as heterosis or hybrid vigor. Remember that these hybrids can be heterozygous for one or more traits, not for all the traits. They can be heterozygous for one or more traits. For example, here we have shown they were heterozygous for the flower color. Then we have got pure line. What do you mean by pure line? Pure lines are simply the individuals which are genetically identical and they breed true for the trait. Pure lines are individuals, progenies, which are genetically identical and they breed true, which are genetically identical and breed true for a trait. Suppose now what are pure lines, how these are produced? These pure lines are produced from a homozygous self-pollinated plant, produced from self-pollinated single homozygous plant. Or two identical homozygous parents. I will explain like this. For example, we have a plant with round seeds. We have plant with round seeds, and obviously their genotype is capital R, capital R. These are round seeds. If by means of self-pollination, this is a single homozygous plant, by means of a self-pollination, we'll go for the self-pollination in this plant, in this homozygous plant, which is homozygous for the character seed shape. This is homozygous for the character seed shape. And we will get what? Plants with the round seeds. These are plants with round seeds, So this is what is called as pure line. This is pure line. So we can say that these are flowers. This is flower, bisexual flower. So this is a homozygous plant. This is homozygous for a particular trait, that is round seeds, shape of the seed. Now we'll go for self-pollination in this plant, and we'll get offsprings, we get progenies, and these progenies are genetically identical. These are genetically identical to their parents. Genetically identical. We have got capital R, capital R, capital R, capital R. These are genetically identical to their parents. They are also genetically identical to each other, and they always breed true. That means these plants will always produce seed shape which is round seed. This plant will produce round seeds. These plants will produce round seeds. So we can say that what is pure line? Pure lines are individuals. These are those individuals which are genetically similar. This is genetically similar to this. This is genetically similar to this. This is genetically similar to this, and they always breed true. They always breed true means that they always produce offspring, progenies, which are genetically identical to them. That means in this case they will produce progenies which would be having the same character generation after generation. That means they would be having the same character, means they would be having the round seeds when they will go for the self-pollination. Or we can say that we have got this flower. This is white flower, purple flower. This is a pea plant with purple flowers. Pea plant with purple flowers. This is purple flower. We'll go for a self-pollination in it. This is a homozygous. This is homozygous plant because both the alleles are similar or identical. We'll get the progeny. That progeny would be having the purple flowers. So this is pure line. And their genotype would be identical. Capital P, capital P, capital P, capital P. So pure line individuals are genetically similar to these pure line individuals. They are genetically similar to their parents, and they always breed true. These pure line individuals, they show the same trait generation after generation because of the fact that these pure lines have homozygous condition. These pure lines have homozygous condition. Sometimes pure lines can be having what? They would be having the recessive trait also, but that should be homozygous. Now, how this was regarding the self-pollinated single homozygous. Now, two identical homozygous plants. For example, we have this pure line can be also obtained from two identical homozygous plants. Two identical homozygous plants. For example, we have pea plant. Pea plant with we will take a pea plant with round seeds and cross with pea plant with round seeds. This is capital R, capital R, capital R, capital R. That means pea plant with round seeds, pea plant with round seeds. Obviously the progeny would be, this is pure line, this is pure line, F1, this is pure line, that pea plant with round seeds. So pure line can be also produced from two identical homozygous plants. That that means these homozygous plants, they have two identical alleles similar. This is the case. We have produced the pure line from the two homozygous parents. This is one parent, it is having this genotype, another parent having homozygous genotype. This we get the offsprings or progenies which are identical to their parents, and they always breed true. That means this individual will produce round seeds generation after generation, in the F2 generation also. So this is what is called as pure line. This pure line selection, it was used by Mendel in his hybridization experiments. What do we mean by pure line selection? In pure line selection, what we do, we take a single homozygous self-pollinated plant, and then after self-pollination, self-fertilization, seeds are collected from this plant. From single homozygous self-pollinated plant, seeds are collected, and these are sown, planted, and individuals are obtained from them. Then these individuals are planted and evaluated for the desirable character. The best progeny is then released as the pure line. Remember that these pure line progenies, these are genetically identical to each other, as well as they are identical to their parents. And sometimes there would be also variation, and the variation in pure lines or homozygous individuals is solely due to the environmental factors. That means if we have got, this is a pure line, this is a pure line individuals. There are, for example, 100 individuals in this pure. These are pure line progenies. Pure line progenies. These pure line individuals, they are all identical, they are all similar, they have similar genotype. Now if there would be some variant, if there would be some variation in an individual, but that would be solely due to the environmental factor. That would be solely due to the environment and not by the genetic means. So we can say that in pure lines also variation can exist, but that is solely due to the environment. And this was regarding the genome, hybrid, and pure line. Thanks.